Hi, in this video I'd like to show you a little bit about DNA topology and how you can simulate it using string. The first topic I'd like to talk about is the major and minor grooves of DNA. So individual strands of DNA are not symmetrical. They have the phosphate backbone, they have the sugar, they have the base, and the strand isn't symmetrical. So when they form this double helix, the double helix isn't entirely symmetrical either. And one of the ways that it isn't symmetrical is it has a major groove and a minor groove. So one side of the helix is more tightly pressed together than the other side. If you make a helix out of string, it will also have two grooves, but they will be the same size. So here I have two strands of string. Each strand of string represents a strand of DNA, and I've marked here one side of each string I've marked green, and the other side I have left blank. And so we can see the origin of the major and minor groove. So the major groove is going to result from one of these sides. It's going to turn into the major groove. The other side is going to turn into the minor groove when we twist them together. So try to keep the green on the same side here. And so when we twist it, we can see that there are two grooves. They're the same size on string, but there are two grooves on the string. And so if you follow the green groove, you'll never reach the white groove. And if you follow the white groove, you'll never reach the green groove. And so the major and minor grooves in DNA are also separate. They're distinct grooves. Linking number is an important concept in DNA topology. Linking number is the number of times one strand of DNA wraps around the other strand. If the one strand wraps around the other strand in a right-handed direction, if it's a right-handed helix, then it will have a positive linking number. For example, this bDNA here it's a right-handed helix, and you can tell that because the front strand passes up and to the right in front of the rear strand, and it has a positive linking number. So this left-handed helix, this zDNA, has a negative linking number. Okay, to show linking number and how to change linking number, I have, again, two strands of string. Each one represents a strand of DNA. This time I have one colored green and one colored white. And so the linking number is just the number of times one strand wraps around the other one. So we can increase the linking number by twisting them in a right-handed direction. So if we take our right hand and twist the string in our right hand towards you, this will make a right-handed twist in the DNA, and it will increase the linking number. So this string now has a positive linking number. And if I were to twist it the other way, if I were to take twist the top of the string in my right hand away from me, or the top of the string in my left hand towards me, we can cause a decrease in the linking number. And so now this string has a negative linking number. Relaxed bDNA has a positive linking number, meaning that without adding any energy, two strands of DNA will just naturally wrap around each other at the rate of one turn every 10.5 base pairs. String, on the other hand, if you just hang two pieces of string next to each other, they generally won't wrap around each other very much, so their linking number, the relaxed linking number, is about 0. So string in its relaxed form doesn't have any linking number at all. Its linking number is 0. The strands don't wrap around each other, whereas DNA would have a positive linking number. When DNA has a linking number other than its relaxed state, then there will be tension in the double helix. So for example, if you wrap it tighter than its relaxed state, it will be overwound, and that can cause positive supercoils. If you wrap it looser, so if you wrap it in a left-handed direction, it will be underwound, which will cause negative supercoils. And in cells, most supercoiling is negative. Most often, DNA is underwound. It has a linking number of less than the number of base pairs divided by 10.5. So from the relaxed form, we can overwind the string just by winding it in a right-handed direction, or we could underwind it from the relaxed state, the parallel state, winding it in a left-handed direction. Like that. So if you twist string or DNA far away from its relaxed state, it will have a tendency to form supercoils. And where supercoils come from is when it's in this tense state, this non-relaxed state, the ends, well, they want to rotate. There's force on the ends to rotate it. So if I just let it go here, it'll unwind. But if I have the ends fixed, so the ends can't rotate, if I'm holding the ends here, that same tension is going to apply to the middle. And so if I give it a little slack, it's going to supercoil around itself in the same way that this free end would rotate, except now it can't rotate without wrapping around itself. And so that's where supercoils come from. And you may wonder why in eukaryotes, since eukaryotes have linear chromosomes, they actually do have a free end like that. So why doesn't it just unspin like it does with string? And the reason for that partially is because DNA is much longer than it is around than its diameter. Its length is much higher than its diameter. And so there's actually friction between the DNA and the water that it's sitting in, and the nucleoplasm that it's sitting in. And so it's just like if you were to take a string or a rope, a big rope, and lay it out on the ground and try to pick it up and rotate it from one end, you'd find that you'd have a difficult time rotating a rope that's laying on the ground because there's friction between the rope and the ground. And that's how with DNA there's actually friction, quite a bit of friction between the DNA and the nucleoplasm. And it's also bound to proteins, which also prevent it from rotating. One other thing I want to point out here is that the handedness of a supercoil will be in the opposite direction of the original coil that caused it. So for example, here I have this overwound string. It's wound quite far in the right-handed direction. So when I let it form supercoils, it'll be trying to rotate in a left-handed direction against the coil, the original coil that I've made. And you can see that, hopefully you can see that this coil here is in a left-handed direction. The one in front is passing up and to the left. But this is still called a positive supercoil because it was caused by overwinding, by a linking number that is passed in a positive direction past the relaxed state linking number. Supercoiled DNA is more compact than relaxed DNA. And that's one reason why in cells most DNA is supercoiled, because the DNA is a very long molecule, and so to compact it so it fits nicely in the cell, it's necessary for the cell to supercoil its DNA. Experimentally, you can see that supercoiled DNA is more compact than relaxed DNA by running a gel. So smaller molecules will run faster on a gel that runs further on a gel. You can distinguish relaxed DNA here from supercoiled DNA. And this can have implications, for example, if you're isolating plasmids from a bacteria and running it on a gel and expecting to see a single band. Oftentimes you'll see multiple bands because different individual units of plasmid will be supercoiled to different degrees. And so you might be looking for one band but actually see multiple bands, which can be confusing if you don't know what you're looking at. Supercoiled DNA is more compact than not supercoiled DNA. Here I have two strings that are the same size. One of them is supercoiled and one of them isn't. And you can see the supercoiled one is more compact than the other one. And this is true of linear as well as circular DNA. Linking number is made up of two components, twist and writhe. Twist is the number of helical turns, so it's the number, literally the number of times one strand is wrapping around the other strand of DNA. Writhe is the number of times the double helix crosses over itself as a supercoil. So when a supercoil forms, for every supercoil that forms, the twist actually moves in the opposite direction. That's how supercoiling restores DNA to its relaxed state, is because the twist in the relaxed state, the twist equals the linking number. If you increase the linking number to restore that relaxed state twist, the DNA will tend to increase or change the writhe. The double helix will wrap around on itself. The only way to change linking number is if a free end rotates or passes across another end. This could be a single strand break, or it could be a double strand break or a chromosome end that wraps around or turns. Generally, changing linking number is accomplished by topoisomerases. It generally isn't accomplished by chromosome ends turning. So the linking number is equal to the twist plus the writhe. So the twist is just the number of times that one strand wraps around the other one. And so in this instance, the linking number, and when I have it straight, there's no writhe in this, there aren't any supercoils. So the twist is equal to the linking number, and the writhe is equal to 0. Now if we let it form supercoils, we can now see that the twist is actually decreasing. So these two strands are wrapped around each other pretty tight, but it actually gets looser as it forms these second order coils. So the linking number is the same. For every second order coil that forms, one of these first order twists is undone. Because of that formula, linking number equals twist plus writhe. That's why even though these are left-handed, they're positive writhe, even though they're left-handed. And that's to preserve that formula, because they are induced by positive overwinding. So there's two kinds of topoisomerases. There's type 1 and type 2. A type 1 topoisomerase makes a single strand cut, so it breaks a single strand of DNA and then allows that strand of DNA to wrap around the other strand in a way that reduces tension. Type 1 topoisomerase does not use ATP, so it can only be used to relieve strain. It can't be used to increase the strain on DNA. So generally that means making the DNA closer to its relaxed form, because it's not using energy, so it can't do anything that would require energy, such as wrapping the DNA more tightly than it's already wrapped. So what a type 1 topoisomerase does is it will make a single strand break and then allow the DNA to unwind. So I have this taped here, so this is overwound, but if I just unattach the tape, then you can see that it'll naturally kind of unwind without me having to add much energy. And then I just tape it back together again, and you'll see, so I just let it do that, and then you'll see that it is more relaxed, closer to the relaxed form, if you just, pull it apart and put it back together. And that's exactly what topoisomerase 1 does. Type 2 topoisomerase, the other kind of topoisomerase, makes a double strand break and passes the unbroken strand through the broken strand. And this uses ATP, so it can actually move the DNA further away from its relaxed state. So it can either add or relieve tension. Topoisomerase 2 is a little trickier to demonstrate, but we'll give it a shot here. So what topoisomerase 2 does is it actually makes a double strand cut and then passes the cut strand over the other strand. And so here we can see we're starting out with a linking number of 4. We can see that this green strand passes over the other one 4 times, 1, 2, 3, 4. And if we let it form a supercoil here, just like that, if we let it make a supercoil, and then, so we got a supercoil here, and then we pass the strands through each other, and then tape it together. We can see that we now have a linking number of 2. So it's now passing over the other one 2 times. So by the action of topoisomerase 2, we have changed the linking number by 2. In eukaryotes, a DNA compaction is accomplished through nucleosomes, through histones, which are the proteins at the center of a nucleosome. Nucleosome means it refers to the histones here and the DNA that's wrapped around them. The histones are positively charged proteins, which means that they will interact with the negatively charged DNA. See the negative charges on the DNA backbone here. The wrapping of DNA around a histone core to form a nucleosome actually introduces strain into the nearby DNA. So this strain can then be relieved by topoisomerase, which actually results in a decrease in the linking number. So even though you don't see supercoiling here, you do actually see supercoiling, but it's solenoidal supercoiling. It's supercoiling around this nucleosome. So if this pen is a nucleosome or a series of nucleosomes, we can show how DNA would wrap around a nucleosome and what that would do to the topology of the DNA. So you can see I'm starting out here with a linking number of 0, and I'm just going to hold on to the string the whole time, so I'm not going to allow the ends to rotate. So you remember the only way that the linking number can change is by the action of a topoisomerase, so if there's a single strand break or a double strand break, or if the ends rotate. And I'm not going to allow either one of those things to happen as I wrap this in a left-handed direction, which is the way it would be wrapped around a nucleus or around histones in a cell. And so I'm just going to wrap this around. And you'll see that what's going on here is we're getting twists. So over here, this DNA has twist at the end, but if I pull it off, you can see that the linking number is still 0. And so the way that a nucleosome contributes to supercoiling, we have, this is a, these here are called solenoidal supercoils. They don't look like supercoils, but they actually are. And then if I let a topoisomerase act on this twisted DNA out here and let it restore it to the relaxed form, so if I just let it unwind here, you'll see that when I pull it off, I actually have twists now. And so that's what happens, that's how nucleosomes introduce supercoiling into DNA. If the DNA wraps around the nucleosome, it creates tension. The topoisomerase will relieve that tension, and then you'll end up with DNA that is away from its relaxed form, and it will have these solenoidal supercoils. Helicase is another protein that can change the topology of DNA. What helicase does, is it splits the 2 strands of DNA. And when it does that, it actually introduces supercoiling in front of the helicase. And this supercoiling has to be relieved by a topoisomerase, a type II topoisomerase, which in bacteria, or more correctly in E. coli, is gyrase, is the name for that type II topoisomerase. So the last thing I want to demonstrate here is what helicase does. So what helicase does is it will separate the 2 strands of DNA, get that, and then it will move along in one direction, And you'll see that in front of the helicase, the DNA is becoming more tightly wound, and behind it is becoming less tightly wound. And so a topoisomerase needs to come in front of it and relieve that tension.